Hello, everybody. I hope you're staying well in these crazy, uncertain times. It's really nice to see you. And I would like to say thank you so much for the invitation to present our work to you today. It's really a pleasure to be speaking with you all. It's been a long time since I've seen lots of you, and that's sad, but hopefully here's to the future, and that we can see each other in person sooner rather than later. I really wish we could have been at Dalhousie though, because like that's where I graduated from university. I would have loved to be in Halifax with you all at the moment, but this will do. We're reaching more people probably this way from all sorts of different time zones. So again, thank you again for the invitation. And what I'm going to talk to you today is about mixtures and multiple stressors, and how can we incorporate suborganismal processes into the DEB framework, and how that will provide a path forward to dealing with mixtures and multiple stressors. And what I want to do right at the beginning of this, so I didn't start off as a DEB modeler, and I still sort of feel a little bit on the outside of it. I'm getting more, and I'm collaborating a lot more with many of you, and it's really exciting, and I love this community a lot. But what I want to do is I want to show the thought processes that I went through, and a lot of people who are also think similar to me, how we got to embracing DEB in our research. So I'm going to start off with a little bit of history and then show you and lead you up to where we are today. And this goes well before my time too, is how we used to do contaminant testing. Remember, it's usually done with this dose-response relationships where we determine a median lethality of a substance. It's like a kill 'em and count 'em method, essentially. And it was tested on a limited number of species that were suitable for laboratory testing. And it's usually based on lethality or some huge gross morphology or phenotypic change. However, things started to change. And so what happened, it started in the 60s, where we started to look at DDT effects. We were noticing that DDT had harmful sublethal effects, that it wasn't killing a lot of birds, especially overtly, but it was causing these sublethal effects, which was causing population collapse, which was the focus of Rachel Carson's Silent Spring book back in the 60s. Coincidentally, she was influenced by research done at MSU, where it was a researcher, Wallace, who noticed that all the robins were gone off of MSU's campus and linked it to DDT. And that's really sort of triggered the book. It's part of her foreword, where she talks about how there were no birds singing. Can you imagine a world with no birds singing? Okay, so she started it. It started actually, this book sort of has been credited with triggering the environmental movement in the US. We had the EPA that formed in the 70s, etc. And then what happened in 1996 is we started to see people started documenting this phenomenon of endocrine disruption, right? So this started in the late 90s and into 2000, where we're starting to see this idea of endocrine disruption, which are all these sublethal subtle effects that were occurring in wildlife and fish and people. It wasn't causing some median lethality. It was causing these subtle effects that could have long-term population impacts. And it wasn't really until the 2000s until we decided to call it an endocrine disruptor and started to refocus on that. All right. And then what we were noticing by, like the traditional toxicology tests that we were using were not effective. I mean, where we have them, again, this median lethality, the kill 'em and count 'em, we have a dog dosage, a medium of number of animals showing a response. But what's the ecological significance to this? This type of approach is hard to incorporate things like growth and reproduction and development and behavior. And how do we incorporate those and bring it into ecological risk assessment? And so what we realized I guess, has collectively realized is that this is a huge problem. It's a gigantic. We've been doing this median lethality types of testing for so long, but we're realizing that there's over 86,000 chemicals listed on the Toxic Substance Control Inventory by the EPA, and really only a handful have been adequately tested for these sublethal effects. And this doesn't take into consideration things like degradation products, mixtures, and interactions. And most of the focus has been on the human health effects after some sort of consumption. But we don't really know what are the effects on fish, wildlife, invertebrates, plants, algae, fungi, microbiome. And what is really hard is this endocrine disruption is subtle, and we haven't even really characterized all the effects that it has. Right? So that's when I started thinking about this problem, was right around this time. And one of the things that I thought, so this endocrine disruption is incredibly subtle. Basically, what we're doing is we're picking up changes in hormones, right? But changes, what do you do with a change in a hormone level? It's really hard to imagine what you do with that. And so what we have to do is we have to convert it into something that shows an effect that could have implications for population level impacts, population risk assessment. So the first step was to try to link these like changes in hormones, because it's endocrine disruption, to something like egg production. All right, and so that's just was the focus of my dissertation, like way back when, where we're kind of like looking at like physiological models. We're starting to develop these where we're trying to, this was a very crude sort of Frankenfish, where we're just putting all these parameters together, a series of ordinary differential equations to sort of simulate the normal hormonal processes within a fish that would be linked to vitellogenin, that could be then linked to the number of viable sites. And basically it was a very simple model with a number of state variables that represent some of the hormones, and then the output was vitellogenin, which then, assuming very little degradation, could be then converted to like a gonad mass or something like that, or the number of eggs. Okay, so that's where we want to, and then right about then, there was this National Research Council in the US. We had this toxicity testing in the 21st century. A vision and strategy came out, and this recommended a paradigm shift in toxicological research. We wanted to transform toxicity testings from these systems based on whole animal testings to one on in vitro methods. We want a new toxicity testing system that looks at biological pathway perturbations, toxicity pathways. And we want to use a more computational in vitro, high throughput tests, cells and tissues, and let's get away from whole animal testing. So that's what came out, saying that we haven't been doing a good job with toxicology. We've been missing all of these subtle endocrine disruption effects, right? And then about 2009, there was a Pelston workshop that tried to take the NRC document. This was sponsored by CTAC. It tried to take the NRC recommendations and try to think about how we could do this in an ecotoxicological context. And from that, I guess there were two challenges that sort of were hard. They impede our ecotoxicology's transition to this 21st century paradigm, basically. One was we have to establish these credible links between responses measured at the cell or tissue level to adverse outcomes that are measured at the whole animal or population level. And then also extrapolation. We have to develop these biologically based quantitative extrapolation tools or models that let us take these cell or tissue level data to individual level data and to entire populations. And so this is where we decided to focus right here, is at these linkages. And from this, what happened is this adverse outcome pathway was born pretty much out of this concept. I mean, it's not necessarily new, But at the same time, what was new was was an organizing framework, putting new terminology on things and trying to embrace the global community to get on board with it. And basically it's a framework that that organizes information along levels of biological organization so that you can make these causal linkages between them, and so that you can then take things that measure at the molecular level, cellular level, and try to infer a population response. Okay, so this is the adverse outcome pathway framework itself. It tries to take all of this information and organizes them into key events and key event relationships. But where you want to get to with this adverse outcome pathway are these quantitative adverse outcome pathways, where you have all of these potential molecular initiating events and key events on levels of biological organization that lead to some sort of adverse outcome at the organismal level. But then you want to take them, and the idea is put some math behind it and try to create these key event relationships from like subsets of this data to get some adverse outcome predictions. And so then you can then link, you can get the environmental exposure that then through some of the toxicokinetic models can then be incorporated into this and trigger molecular initiating events. And so then by the combination of having the exposure modeling plus the risk characterization, you can get well, basically this gets you the hazard, and then you have the exposure, and the combination of both of those things gets you to the risk. Okay, so this is where we want to get. But to get to this, and this is an approach that's been embraced quite a bit, it's challenging to get to these. This is where we want to be, but to get there requires a lot of data collection. And so that very simple adverse outcome pathway that I created for my vitellogenesis model is evolved and changed. And so here's just an example to show you. Right here, like for example, they've taken the fathead minnow, the EPA scientists and collaborators have taken the fathead minnow and created this really, really detailed vitellogenesis model, essentially, from it, trying to get everything that they can into it. Then that model, so you can take like some sort of chemical like fadrazole, causes an inhibition on the aromatase. You can then get a prediction of vitellogenin production from that. So this is a model that will predict vitellogenin, right? From that vitellogenin production, you can then put it through the statistical model where you get oocyte growth dynamics. And from that oocyte growth dynamics, then you can then link it to another population model where you have a declining trajectory. How much does that, what concentration of that fadrazole will cause a decline in the population? So there's these three separate models that have been linked, and it's quite an amazing amount of data that went in to do this. It's quite a lot of work, right? But it's hard. This is hard to do. This is hard to get all of this data. I think it took over 10 years to get all of this together and it was probably quite expensive. All right. And so then we take a look at, so it's amazing data set and it's great. I think it's fabulous. But there's also some limitations, I think, as well. And if we take a look at this adverse outcome pathway, how it's structured, it seems pretty linear, right? And so were those models. One links to another, which links to another, to another. It's very linear, and it will provide some information. It'll provide, and especially if this is linked into a broader AOP wiki and networks and stuff like that, that will help feed things. But this as it stands is kind of a very linear. The analogy I make for it is kind of like if you have this great big black box and you have a flashlight and you're putting this narrow beam, and that's what you're illuminating. You can collect all of these beams and start to put them together with networks and stuff, and that is one good approach to doing it, and that will give us lots of information, especially with a common framework where we can structure all of this data. But it's time consuming and it's going to take a long time. But I do think there is a lot of value in that approach. But it got me thinking, okay, so this is, so again, like I said, how did I get to DEB from this? I started thinking about, okay, well, we have the hormones and we're linking it to egg production, but I got stuck there. So we have egg production, especially with the production of vitellogenin. So we get this big chunk of vitellogenin in the fish. What do we do with it? Well, we have to send it into eggs, right? So it's either the number, but there's a number of things that could dictate the number of eggs, right? It could be the size or the number, and that's going to carve up that vitellogenin chunk somehow, right? So the size and number of the eggs is going, and then it doesn't matter too whether there's environmental stressors around too, but like the size and number of eggs, that's, how do we figure that out? Well, that's a life history, sort of an evolutionary type of question, right? There's some sort of pressure that's put on the population to determine the size and the number. And we've seen some of these talks already. So we have to sort of link this somehow to this puzzle pieces right here. But also too, the environment as well plays a big role, the condition of the organism. It's like all of these are based on organisms that were brought up in the lab under ideal conditions, but we know like condition will affect the number of eggs you're going to produce, all of those things, right? So how do we link the environment and the condition to these pieces of puzzle? And so that's where I started to think about DEB. But also, I just wanted, again, another plug for the adverse outcome pathway. I think that the actual idea of it, even though there's some limitations to it, by putting all the information together, and the way that you can do it lends itself to letting you think about these questions in different ways. And you don't have to approach the adverse outcome pathway from one way. You can start from the bottom up and try to go from molecular initiating events to some genes and up, and you can try to go that way. That's a really hard way to go. A lot of times people will start from here and try to incorporate some of these suborganismal level processes into link it to the individual. That's maybe more of another way to go. That's a top down. And then there's a middle out. So you can start to explore this framework. How do we go from levels of biological organization? And also because the AOP, and you can put all this information together, but also because this AOP is chemically agnostic, you don't need to just look at chemicals. You can look at contaminants can inform the structure of the AOP and how these relationships will flow, but you can look at a bunch of other things to help you inform how all this will work. And so we've looked at a number of different stressors, not just contaminants, and how do we bring in evolutionary pressures? How do we bring in the environment? And so we've looked at a number of different things to try to like interrogate the structure, to try to figure out how do we put these pieces together. And so these are some of the other stressors that we've looked at. And what we want to do with that then, to put these pieces together, we have to think of like these feedbacks. We have to think about life history trade-offs, growth reproduction, egg size number of some examples. And we have to evaluate with different environmental stressors like changes in food, changes in community structures like the predator-prey dynamics, and also changes in temperature. And so that's where I started thinking about the DEB theory. That's how I got to you guys, right? And so again, this is some work that I've done with Laure Peccari. And so basically working with a bioenergetic model of growth, development and reproduction throughout the life cycle of an individual with dynamic food and temperature conditions, but using Koyman's standard DEB model. So like, again, the theory, we know you have all been thinking about multiple stressors, contaminants, hypoxia to implications. But then I really wanted to think about like those evolutionary pressures. So that's what I'm going to look at first, and look at these life trait variability among related species. DEB provides rules for the variability, but it's never been applied to the physiological basis underlying life history traits among stocks of the same species. And I think that can provide a lot of insight into how these DEB parameters can change based on evolutionary pressures. And so what we used for this, and this is still a work in progress, but I wanted to show you where we were going with it. And hopefully by the next DAB, I have more to say about it. But basically what we looked at, we have one species, the lake trout, but they have a bunch of different types of ecomorphs essentially, and they have a bunch of different pressures, and they have different mortality rates, different egg sizes, different growth trajectories. So they almost act like different species, even though they're the same. And so basically we took this, we took the lake trout. We have some this is again from Laura. I got this from one of her talks a long time ago. But basically looking at environmental data. This is what we're dealing with, right? We have our environmental data and the organismal data. These are things that we can measure, the length, weight, and fecundity. We have some forcing variables, and then we have to try to estimate what these state variables are with the DEB. So what we were going to work with our lake trout is we had these 4 different lake trouts species that had very sort of different life history characteristics. They were either short-lived or long-lived, but some of them also in the natural systems had low reserves, like what I mean is lipid reserves, and some of them had high lipid reserves. And there was some theory about when they take in the energy, where do they put that energy? Some of them are going to put it into reproduction, especially if they're short-lived right away, and some will put it more into growth if they're long-lived. And so there's these different things, these different sort of choices, I guess, they make based on their life history. And so we collected data. This was, we had these 4 stocks that were housed in this long-term common environment. So they were all raised at the same environment from hatching. And they had either a high or a low food ration at the hatchery. And we monitored these individuals. We had over 200 of them, and we monitored them for 6 years. Every 6 weeks, we looked at their length, weight, and their lipid levels using this non-invasive fat meter. And we also measured their fecundity when they spawned, and their egg size as well. All right, and so some of the things, some of the early DEB modeling of this species, like we're making this assumption that the stocks are going to differ in parameter values that can be related to life history, because we were seeing changes within these stocks based on the DEB modeling that seemed to fit with their life history. Like the manitous are short-lived, Michipicotes are long-lived, and then the fecundity changes as well. And so we want to keep exploring this a little bit more to sort of relate those. We also have another type of morph we're also looking at, which is a ciscoetch, which is completely different than these lean types. But we're going to keep moving on this to try to see if we can link some of these DEB parameters to some of these life history characteristics. So again, so that's linking. This is maybe a step forward to try to get some of this evolutionary pressures incorporated into this sort of puzzle piece, these frameworks. And DEB is a really useful pivot. Now, to bring the environment and the condition, of course, as you all know, we need to, I think DEB also works as a really good pivot for that. And so this is where the rest of my talk will be, is how we bring in this stuff, the environment and the condition into this AOP. This is what I call the AOP framework right here. And to do this, I didn't do it by myself, that's for sure. I've been working with this wonderful group of people through the Nimbus working group, the modeling molecules to organisms, that was sponsored by the NSF Foundation, National Science Foundation. And so all of these wonderful people have contributed a lot of time and effort into this. But basically what we were doing here is, again, we're going from molecules, cells, organs, individuals, populations, communities, and ecosystems here. We have these AOPs that can be linked to the DEB models, and then we have the DEB that can go to the ecological models. But again, this individual is the pivot, and you can see the common denominator right here is DEB. So the key is to get the AOP linked to the DEB. All right. So there's a, like I've mentioned, there's, again, that's where I come from, is this quantitative adverse outcome pathway world. There's a lot of pros to it. It can accommodate all these high throughput testing and minimize animal use, and it is quite mechanistic. But the cons are, the ones that have been described are pretty linear. They're starting to get into networks and things like that. But they always converge on a single adverse outcome, like growth or reproduction, and they don't link them. They're calibrated for ideal lab organisms under ideal conditions, and they generally require quite a bit of data. The dynamic energy budgets to link, again, they have these energetic trade-offs. They can handle that between the physiological processes. They can be applied to different environmental conditions, which is where we can bring the environment into the framework. They can be applied to different life stages and species. But they're the cons, of course, which is a bit of a barrier, is these abstract, that fantasy world, where we're trying to link these abstract variables to suborganismal processes. It's hard to do. And it does need some specific data to parameterize as well. I know the AOPs require a lot of data. The DEBs do as well, it's just different types. Right? However, we can make these connections, the AOP DEB potential, if we can link these suborganismal processes to energetic models. We have the potential to go from like thousands of chemicals and stressors to millions of species, right? I know that's ideal. That's what we could do. I mean, especially with your Add My Pet, where you're adding more and more species to that every day. We also then can make it more realistic, because then we can predict the effects under varying environmental conditions, food and temperature, etc., and multiple stressors. And so this is the conceptual model that we laid out from our group, where we have all of these AOPs that are integrated by the dynamic energy budget model that can be scaled up to the IBMs or population matrix models, etc. So how do we do that? So I know Roman Aschow, Shaw and Yager have talked about these physiological modes of actions, and we agree as well that the physiological modes of actions are key to linking these adverse outcome pathway key event relations to the DEB. All right, so basically you have these key events that link into these DEB fluxes, into these physiological modes of actions, right? So just to think about this more from an AOP. So again, like this is what the AOP type of data that you collect. You know, you have across levels of biological organizations. You have this PPAR alpha assay, you know, that goes up to a muscle protein catabolism. And that with that, if you could link that to the DEB processes, the physiological modes of action within the DEB. Similarly, but it doesn't have to be just one, you know, mode of action, one AOP. You could probably potentially link another one as well. All right. So theoretically, this seems like a really good approach, with the DEB sort of in the middle, integrating all of these responses. However, there's a problem with it. There's no unique connections from these real processors or flows to these more abstract DEB processes. Again, so this is the problem. So this is what our group dealt with. Like, how do we deal with this? And so we came up with, I guess, three different options to deal with it, and I'm going to talk you through them right now. The first option, and I'm not going to go through this one in detail because Eric Mueller has presented about this to you all before, but basically what this one is created, what he did was change the energy allocation rules so that it can accommodate some of this data that we would collect from suborganismal levels, estradiol, little agenin, all of these things, and incorporate that basically have a demand DEB, so you have a feedback of the reproductive buffer on the reserve partitioning. And it seemed to predict the empirical data quite well, right? So that he's presented on that before, so I will let you read up on that if you haven't read about it yet or heard about it. The second option was to add a reproductive hormone module to the standard DEB. So basically we're taking our quantitative AOP and then collapsing it down into something simpler with a regulator, active regulator, and activator. And we have that signal that goes into the synthesizing unit of the DEB to predict the reproductive matter, right? And so Dina has actually presented that to you all, I think last time, So I won't go into too much detail there, but that's one way we can take we just collapse all that complicated quantitative AOP framework stuff into something much more simpler and put it in here. And so Dina's done that, and we're still working with this though. We're trying to go even simpler. We have a project with sea lamprey parasitism. Essentially, we stuck sea lamprey on fish, lake trout, and this is my PhD student, Tyler Fergus's work. It's his PhD work. He stuck sea lamprey on lake trout and let them feed for 4 days and then removed them and then monitored them for about a year. And what he found was like a very severe parasitism attack, basically, which is what we call a type A wound. It's like a wound that actually penetrates the muscle. What that happens is there's an increased likelihood of skip spawning as a result of that. But also that it's not quite as clear cut. Like the lipid levels before parasitism also contribute to that skip spawning event, but also estradiol concentrations the year after also predicts skip spawning. So we're taking the egg loading module that Dina's worked on, and we're trying to collapse it to something even simpler, just to having an estradiol feed, and to try to, which will link into the condition and the reproductive buffer of the fish. And we have a big data set for this, and so we're going to try to incorporate this to even be a simpler type of approach. And that's in progress by Tyler Fergus. We should be wrapping this up pretty soon, probably in a few months. And so you can hear it from him when he's done. Okay, but it's pretty cool. So he's working with Dina to incorporate that into the DEB model. Now, finally, the last one option, which you probably not have heard from us yet. And so this is still a bit in progress, but it's getting pretty close to being published, is to statistically link our omics signatures, our omics, like the transcriptomics and proteomics, to DEB physiological modes of action. Okay, so this is was a big project from our working group as well. And basically what we were going to look at is a mixture. We looked at a coal ash mixture exposure study. So this was based on, so like in 2008, the largest coal ash spill in North America occurred at the Tennessee Valley Authority's Kingston fossil plant. All right. It's the largest one. And there's a lot of cleanup, of course, associated. And then there was some monitoring of it, and the data that came from it, where there was like mixed and inconsistent effects on fish and invertebrates. And so what we decided to do, because we were just starting to work together as a group, we decided to conduct a Daphne experiment with this DEB framework in mind to see if we can pick up any sort of unusual signatures, etc. And this was done by Terry Matthews at Oak Ridge National Lab. With a lot of us contributing. And so basically what we looked at, we had two different rations, high and low food, and we looked at Daphnia, and we conducted it over a 31-day test. We looked looked at a high food ration and a low food ration. And basically what we found was that survival, there was variable effects with survival only with the low food, and it was different than you would expect. With the no ash being right here, the high ash had the highest survival under the low food ration, and the lowest food and the lowest ash had the lowest survival, which was kind of interesting. But the coal ash is a mixture of mercury, selenium, arsenic, all sorts of other types of chemicals. So there's these things that are competing. Also saw that the neonate production were a little bit different with the high food and the low food, and the average length, as well as the age at first clutch. So basically, the ones that took forever to start to produce their first clutches were the ones on the low food controls plus the low food and the medium ash, but the ones with the high ash levels all seem to speed up their growth to produce their clutch at a younger age. That was more in line with the high dose, with the high food levels. So you can see that there's something going on here, but we need to figure out what the mechanisms are. It's really hard to figure out what's going on exactly with this data. So we thought this was a particularly good example to explore how moving our omics, if we can get some omics signatures and relate it to DEB. So basically what we want to do is we take omics samples from a standardized test and relate it to these physiological modes of action, which then can of course be then interpreted for a population model and effect. So to do this, to link the omics to the DEB, we modified that, we adapted that. We learned a lot in that Matthews experiment, so they adapted it basically by broadening the doses. So they used at 6 doses from no to low effects. They looked at 1 food ration only, took 4 sampling points through the life cycle. So these are the sampling points, day 6, 13, 17, and 24. And from actually these 4 sampling points, they had to, and we had to be very careful about when we exactly sampled at what time of day and at what stage the Daphne was. And we needed to get enough data so that enough tissue so that we could do the transcriptomics. So we then took 4 individuals and individually sequenced them, looked at the differential expressed genes, and then KEGG pathways, some bioinformatics, and some data DEB modeling. And so this was done by this group of people here. A lot of work's gone over the past 6 years into this. And so we have Louise, Phil, Andre, Leah, Teresa, Natalia, Roger, and myself, all part of the Nimbus group. And so here's some of the empirical results we got from it. So we see there's these subtle effects that are happening on length through time and the cumulative neonate production. All right, you can see that there are these different doses of, we standardize it to mercury to do this mixture effect. It is a mixture of things, but we use the concentration of mercury to set up our doses. So we have 6 different doses. And then from those individuals that we took from the experiment, they did the RNA-seq and look for differentially expressed genes using a couple of these bioinformatics tools, which Phil can probably talk a lot more about than me, like EDGE-R tools and enrichment analysis. And then also in the KEGG pathways, in addition, this DR omics analysis was done, and this tries to identify significant responding genes to the dose of the chemical or the stressor. It uses benchmark dose, basically, which is a dose or concentration that produces a predetermined change in the response of an adverse effect. And so we use this BMD, well, Phil and Leah use this BMD-ZSD, which is essentially the concentration at which a level change occurs compared to the control, given by a factor times the residual standard deviation. And that Z value defines the factor, and it's usually set to 1, which is what we do. And so this is what we get. This is just to show you an example. There's a lot more came out of it, but what we find is the most significant responding genes over this coal ash concentration have a benchmark dose that's below or equal to 0.06 0.06 nanomolar of mercury. So very, 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 very low dose, which is much significantly lower than the phenotypic effects that we observed in our previous study. All right. And then, of course, we have the omics and we have the genes, the differentially expressed genes, and we also need to relate it to the DEB. So we have to develop the model for the DEB, and they use this. So Andre used the standard DEB model with the TKTD model and a scaled internal concentration with body size feedbacks, and then we scaled the internal concentration. It was used to calculate stress level and explored 6 different physiological modes of actions: decreased energy conduction, decreased feeding, hazard, increased allocation to soma, increased cost for growth and reproduction, and increased maintenance cost. And this DEB model mimicked the experimental conditions. And the goodness of fit was calculated using like the mean relative error and symmetric mean squared error for each of these physiological modes of actions. And he took a bunch of, from the simulated models, could take all of these daily outputs that we could link to the omics, like stress level, structural length, etc. You can read that all here. And Andre can talk much more is much more informed about this than me. So questions about this, you can ask him. Okay. So the omics to the DEB, so we have the DEB model, and now we have the omics, and we have to link them. So the parameter values of the physiological modes of actions will match to the omics through time points and coal ash concentrations. So we looked for genes that were predictive of parameters within these physiological modes of actions using some variable selection approaches. There's one that's called the lasso regularization method, where there's a penalty on parameters based on Manhattan distance. And this other one, which is the arachne, this is used, I've seen this used quite a bit as an algorithm for reconstruction of accurate cellular networks. So it uses mutual information theory to define the networks of interacting nodes. And then these were all sorted and added to the model using these bioinformatic techniques, Curie and Ranger, random forest, right? And so that's what they were done. And so to get to that part, so here are the cool results that they found, essentially. So basically, what you do is you get an R squared from those models. And so here's showing all the parameters for the physiological modes of actions. And this is for the 2 different approaches. There's the lasso and the arachne. And basically this is the R squared, and we want to look at the mean because it kind of is, it sort of takes everything into consideration. And basically, and then these different modes of action, physiological modes of actions, are color coded. So we have increased cost for reproduction, decreased energy conduction. All of them are here. And basically what you're looking for in these, for both of these models, is for the ones with the higher R squared, so the ones that are going into this area. And in this particular one, we see that there are 2, like we have the increased cost for reproduction and the decreased fraction to soma. Seems to be the best performing ones right here. All right, so that's one piece of information that we can use. Okay. What we also looked at was the number of genes that were predictive of these models. And so basically using these 2 different models, there's a lasso and the arachne. So we wanted to look at the number of genes that were contributing to that prediction. And so basically they made the assumption that the ones with the smallest number of genes were probably the better models. And so in this table here is the number, so you want to have smaller numbers of genes, and that's going to be a better model. And so looking at the increased cost for reproduction and the decreased fraction of soma, they have fairly good, compared to the rest of them, they're using a lot less genes, right? In the arachne model, this one does the increased cost for reproduction, uses a lot less genes. This one doesn't quite do so well with the decreased fraction to soma. But so these two models are seeming to do reasonably well. And that's in comparison to something like this increased maintenance cost here, which really has using a lot of genes. And also it doesn't have, this is it here, right here, it doesn't have a very good R-squared value. So we're looking at like two potential physiological modes of action. And then when those are modeled for the DEB, you see here we have the increased cost for reproduction and the decreased fraction to soma. This is by Andre, and you can see that they, the increased cost for reproduction does a great job with the data, much better probably than the decreased fraction to soma. So we're thinking that this one might be sort of the dominant physiological mode of action here. They also explored whether or not you can do combination of physiological modes of action. And so basically they used the increased cost of reproduction, which is what we have here, and they added one other physiological modes of action. Okay. The two approach led, but basically what you see is these two approaches lead to different selections of models. And so it's not as clear cut what's happening here. There could be maybe the DEB models are overfitting or something like that. So there's a lot of variable stuff happening here that needs to be explored a little bit further. But it looks like the increased cost for reproduction is the dominant pathway, and adding these other ones didn't do very much and actually sort of had a poor fit. Okay, but also what was really cool about it is that we have those parameters in the DEB that are associated with the PMO. And what we could do then now is we can link special genes to each of those parameters that maybe give us some sort of biological indication of some sort of biological function. And so this is, so for these are all the physiological modes of action. These are all the parameters in the physiological mode of action. And they were able to link a lot of genes to each of these. And I'm just showing you this as an example because there's a lot more in this than I have time to. But this is just the stress response, and these are the number of genes that are sort of associated with it. And just this particular one right here, this gene pathway, this is identified as a predictor, and this is to the stress response. And this particular one has been linked to oxidative stress signaling in plants. And so it can be induced by different external stressors such as UV exposures, high temperatures, and toxic metals. So it seems to make sense, and it's giving us an insight into the biological underpinnings by some of these parameters. So basically, I'm kind of running out of time, but basically our DEBomics, basically what it's doing is it's combining this unbiased power of the omics with the mechanistic details of DEB modeling. And it can determine the selection of suitable physiological modes of actions, either single or combined, and some molecular insights into the biological functions associated with the DEB parameters and the input data. The selection strategy is unbiased, and it assumes that these key DEB parameters represent biological function and directly associate specific genes and functions to the DEB parameters. And this is amazing. This is great because it helps us set up this mixture toxicity evaluations and a path forward for incorporating a lot of these suborganismal processes into ecological risk assessment. So I'm just going to wrap up now. So basically, hopefully you see now that we've sort of loosely strung all of these things together using DEB as a pivot. Now, we've just sort of started to create those links. Of course, has to be a lot more exploration into these before we can move to the puzzle step 3, I think, and before we even figure out what puzzle step 3 is. But I think we've started to make these linkages, and I think there's so much that can be done. And I just want to bring you back to the scope of the problem. There's 86,000 chemicals. We have mixtures, we have degradation products, multiple stressors. There's increasing restrictions on animal testing. The FDA recently, like in 2019, is going to get rid of animal testing, essentially. We have funding restrictions, and we have millions of species to deal with, right? So there's a lot to be done and a lot to explore with these linkages. And one direction we're going to be going into in a great deal is the PFAS chemicals. These are molecules that contain carbon chains attached to fluorine groups. They've been non-stick coatings, repellents, flame retardants. They've been used since the 1950s. There's so many different congeners. The most studied are two, PFOA and PFOS, and they've shown harmful effects at the parts per trillion range, and they're found in drinking water, which is disturbing. But there's a lot more others. They're found in aquatic environments, and I think they're estimating there's over 9,000 types. They have these chronic sublethal health effects that seem to hit every physiological system. And so this is like a perfect sort of system to sort of explore this. And I think that's what I'm going to be doing for a lot of my career in the next little bit. I'm with our PFAS center at MSU, just to show you sort of how it works. Like this is sort of the systems biology approach and the quantitative AOPs that can link to the pathways. We could do all these high throughput testings and omics, and this is sort of what the data that you can get from ToxCast, where you have the dose here on this axis, and then you have like this right here is this cytotoxic burst. This is the concentration that causes the cell to burst. But there's all of these processes that are affected by this PFOS chemical, this PFOS, I believe, PFOA. Anyway, but you can see here that like at really, really low doses, we're hitting some nuclear receptors, and as you increase the doses, we're getting in more and more processes. And so basically, like I said, we can start to incorporate, as we get a hit on some of these receptors, we use the DEB to integrate it, right? To try to figure out how that mixture or the one chemical or mixtures of PFAS, because they don't occur in isolation, for sure. And so to do that, and so that sets up a framework with DEB providing a path forward in this entire system, where we have the high throughput testing, a lot of QSARs and computational modeling, looking at the pathway perturbation, look at the in vitro dose responses and in vitro to in vivo extrapolations. We incorporate them into the adverse outcome pathways. And we can then look at cross-species extrapolations and life history characteristics to predict differences in sensitivity and susceptibility that get us to this ecological modeling part here. And that's all I have. I have a lot of people to acknowledge over the years, and I certainly do not do all of this myself, and a lot of hard work by a lot of people. Thank you.